Aloha, everybody. Thank you guys. Thank you all for joining us for this Thursday's June 4th Hanama Talks. Today I have Eileen Nalley, and she'll be talking about pollutants on your plate, land-based pollutants in Hawaiian reef fishes. So without further ado, Eileen.
Thank you. Hi, thanks everyone for tuning in. So I work at the Hawaii Institute of Marine Biology, and I'm going to be sharing with you today a bit about some past work that we've done and how that's informing our current ongoing work in Maunalua Bay, as well as future work. So I'm going to be focusing today on land-based pollutants in Hawaiian reef fishes. But this, oh, I'm not able to advance the slides now. Let's see, there we go. So this interest really spurred out of some work that I was doing looking at the diet of herbivorous reef fishes, specifically sturgeon fishes. So as you can see in this image, these fish graze over areas of reef that don't appear to have a lot of food, but they can be in very high densities and at high diversity. So you can have a lot of individuals of a lot of different types of fish eating in one area at one time. And what's particularly interesting about these fish is that they eat, the majority of their diet is comprised of what we call turf algae, which is this fine filamentous algae that covers the reef in a fuzz. And a lot of these fish end up eating a fair amount of sediment in addition to the algae that they're eating intentionally. Others are intentionally consuming a lot of algae. And so these fish are commonly consumed by people and they can be quite long-lived. So this started to kind of drive this question in my mind of whether reef fishes, and especially those that eat a significant amount of sediment, are accumulating land-based pollutants in their tissues. And if so, were they doing it at a concentration that could be problematic for the humans that eat them? And so that's kind of what the driving question behind this work is. And Hawaii is a particularly interesting place to do this kind of work because we're an island. And so in a lot of other locations, you might have pollutants that are transported from other nearby areas, but here we're pretty self-contained. And so a lot of the pollution that we see is really coming from this place, which is both can be frustrating, but is also an exciting thing because it means that there's a lot of opportunity to actually solve these problems by local solutions. And so land-based pollutants can come from a variety of sources. They can come from agriculture, like pesticides, herbicides, fertilizers, things that are used to help plants grow or to prevent pests. They can also come from industrial use. So large urban cities have a lot of pollutants that they put out. Buildings, urban and residential development have a whole different suite of pollutants that might be associated with them, from cars, as well as things like termite treatments, herbicides that people are putting on their lawns. Even stuff like that can impact the environment around you, as well as a significant amount of military development. And all of this is also in addition to what you see in the second image from the left there, which is also just an issue of sedimentation. And so in a lot of areas, the land has been developed in such a way that it doesn't retain sediment during heavy storms. And so that sediment can be washed off onto reefs, and it can carry pollutants with it. Pollutants can also be transported through groundwater onto the reef, or just through surface runoff on heavily paved areas. So you can see that there are a number of different sources and potential root causes for the pollution that we might see on coral reefs. And again, this is particularly interesting in Hawaii because much of our development is along a pretty narrow strip of coastline. And so there isn't a lot of land mass to kind of capture those pollutants. And when we think about pollutants in fish, people are often thinking about mercury and tuna or your higher predators. and consuming too much and pregnancy advisories and things like this. And this is very valid, but there's much less attention being placed on the smaller reef fishes. Yet these fish are commonly consumed and they can have great cultural importance in many locations, especially throughout the Pacific Islands. And so it's very important that these fish also be incorporated into our consideration of pollutants in fish, that we don't solely focus all of our attention on the larger pelagic, commercially important fish, and that we're also considering the impacts of pollutants on these smaller recreational subsistence fisheries that are quite pivotal throughout the Pacific Islands. And so why are we interested in Maunalua Bay specifically? Well, it has a very interesting history as well as a very important role at present. Historically, this is an image of Maunalua Bay from the Bishop Museum archives. And so this is a horse that's drinking fresh water from groundwater outflow areas on a shallow reef. So there's a lot of groundwater being discharged in this area. And so it carries with it anything that was put into the system on land. And so in a more rural setting, this might not be problematic, and in fact, it's beneficial for these horses. But once you start thinking about development and the way that these areas have changed, this could be pretty problematic. In addition, historically, there were a lot of fish ponds throughout Maunalua Bay. And so in this image on the right, you'll see there was Wailupe Fish Pond that got filled in to make Wailupe Peninsula, the Kupapa Fish Pond in Lucas Spring, which was filled in to make Nui Valley Peninsula. We also had a fish pond, Pico Lagoon, as well as further in what is now the Hawaiʻi Kai Harbor. And on the left here, you'll see an area which would be equivalent to over here, where you can see there's hardly any development historically. And so this was a very different landscape relatively recently. And many of these areas have now been dredged and filled in to create residential development. And this makes it particularly interesting when we think about pollutants and their transport onto reefs, because you have a very changed, developed landscape with nearshore adjacent reefs. So this is Maunalua Bay at present. Here is where the old fish pond was. Here's another. Here's Pico Lagoon, as well as Hawaiʻi Kai Harbor, where there was a fish pond historically. And these red arrows point to outflow areas. And many of these are now channelized streams that carry through a kind of concrete channel the pollutants that might be coming from these heavily developed drainage areas. And as you can see in this image, there's a lot of nearshore reef in this area. And this is an area that's heavily used for recreation, both for swimming and boating, but also for fishing. And so it's really important to understand how these changes in development might be affecting the fishing and the people that live in these areas and eat those fish. And so our aim in this project was really to address each of these areas where you'll see these red arrows and kind of understand what the different pollutant signatures are in these areas. Let me find my clicker again. Oh, there we go. And so an important thing to consider is, what do we already know about land-based pollutants on Hawaiian reefs? And how can this inform our work in Maunalua Bay? Because this is very time and labor intensive. And so it's important to think about what's already been done and how can we learn from that work. Well, for the past couple of years, we've also been working on a project to sort of understand baseline concentrations of metals at sites around Oʻahu and Kauaʻi. And so we sampled a number of different locations that vary in their type of land use, ranging from residential to agricultural, harbors, areas adjacent landfills, as well as areas adjacent urban and residential development. And in doing this, we looked at the metal concentrations in sediment and in the tissues of fish. And at each of these locations, we also conducted grain size analysis on the sediment samples to better understand whether areas with very fine sediment had a higher pollutant concentration, which is what we would expect because in many cases, pollutants adhere to the finer sediment particles and get transported that way. So to just kind of give you an idea of some of the things that we saw in this baseline study, here I have four different graphs that have sediment pollutant, metal pollutant concentrations in sediment from a number of different sites. And so on the y-axis of each of these plots, you'll see the concentration of a different metal. Here in the top left, we have arsenic, on the right, lead, bottom left, nickel, bottom right, zinc. And then on the x-axis, we have different sites. And the different colors of the points refer to the expected effect level on marine life for locations that have these metals in this concentration in sediment as set by NOAA. And so at each location, we took 3 replicate samples. And you can see that there's some variability between locations. So to start with arsenic, which was historically used in agriculture here in Hawaii, we see at Maunalua Bay, which is the subject of this talk and our ongoing work, we see really high concentrations of arsenic at levels that exceed what in orange is the probable effect level. And so green is no effect expected on marine life. Purple is a threshold effect level. Beyond this level, you might see impacts on marine life. Dots that are in orange, you would expect to see impacts on marine life when you have the metal at this concentration in sediment. And so it's very interesting that in one of our samples from Maunalua Bay, we see this really high concentration of arsenic. We also saw pretty high arsenic concentrations in Hawaiʻi Harbor on Kauaʻi, which I'll be talking about a bit more in a minute. Looking at lead, which is more of an urban industrial pollutant that can come from a number of sources, including burning things, cars, combustion, we see high concentrations in Kewalo Harbor, which is a heavily urbanized area on Oʻahu. And again, I'll dig into that a bit deeper in a minute, but we also see high concentrations again in Hanapepe, which is a harbor on Kauaʻi that I'll show you an image of and we'll talk a bit more about in a second. Looking at zinc, which again has similar expected sources as lead, we see high concentrations again in Hanapepe as well as Kewalo and interestingly in Maunalua Bay. So that's something we're interested in looking at further. I included nickel here just as an example of a metal that can be present in our rocks here naturally at high concentrations. And so this is something that's important to consider when you do this kind of work. We see several locations with this orange or probable effect level. And so a question that I think about often is when you have metals or other pollutants that are at high concentrations, it's important to think about the source. And so is this a naturally occurring metal or is it from anthropogenic or human sources? And regardless of whether it's naturally occurring, would it be naturally occurring on reefs at this level if we didn't have the issues with sedimentation that I was talking about earlier, where because of development, we have unnatural amounts of sediment ending up on a reef? So it's just something to consider and sort of an example of the complexity of this kind of work and the variety of things that are important to think about as we go through interpretation of results. And so 2 locations that sort of jumped out from that sediment work were Hanapepe on Kauaʻi and Kiwalo on Oʻahu. Both of these are harbor locations, and both have pretty industrial components to them and sort of heavily urbanized areas. Hanapepe also has a large agricultural area as well as an airport and a former dump nearby. And so it has a very complex signature in terms of land use as compared to many other locations. As you can see on the right, Kiwalo is a very urbanized, heavily industrial area that experiences a lot of runoff from cars and buildings and other sorts of development, as well as a former landfill site and a number of other land uses. Sorry, my mouse is pulling things up here. And so it's a very interesting place to consider for pollutants. But again, it was interesting to see these high levels of arsenic that I have highlighted here in Maunalua Bay, because this is an area that is not really industrial, though it is heavily developed and urbanized, but it's something that made us curious for future work. And as you can see, it's one of the 3 samples had this very high concentration. And so it turns out that this is sort of not an uncommon issue with pollution work, because often samples are taken, as you see on the left, in this discrete method where you take 3 samples that are far apart from one another in the same area that you then compare to one another. And so something that we're transitioning to and using in our future work and our ongoing work in Maunalua Bay is multi-increment sampling. And this was suggested by the Hawaii Department of Health as the way that they often do their sampling on land and terrestrial systems. And so basically we make a grid of the area and take a lot of samples spread out over a large area. And then we mix these all together. And we do this again in the lab where we take 50 subsamples from this larger sample. And in this way, we're able to get a more well-mixed representation of the area. And we're not as likely to miss a pollutant if it's patchily distributed, which they often are. And so this is one way we're learning from our past work and implementing that in our current and future work in Maunalua Bay. But the question was really whether reef fishes, especially those that eat a significant amount of sediment, were accumulating land-based pollutants at concentrations that could be problematic for the people that eat them. And so I'm going to show you a series of graphs based on the work that we've already done that we're using to help inform our work in Maunalua Bay. And in all of these, they're similar to the sediment, where on the y-axis, on the vertical component, you'll see metal concentrations for different metals in fish tissue. On the x-axis are the locations or the trophic levels, and by trophic level, I mean, is the fish a carnivore? Does it eat sediment, a detritivore? Does it eat algae, which would make it an herbivore? And in locations where there were similarly high concentrations of that metal in sediment, also, I have an asterisk marked next to that location, which I'll point out. And then points that are abnormally high, I've placed the picture of the fish that belongs to that point next to it, just so you can have a better idea of what that really means. And when possible, I've overlaid management threshold levels. And this is just to give you an idea of what you'll be looking at, so you're sort of oriented to the graphs before we get going. So to start, we'll look at mercury, which is what people often think about when they worry about metals and pollutants in fish. And so this is typically something associated with top predators. And so it was interesting and reassuring that one of our more predatory fish that was included in this study, the trumpet fish, did have the highest concentration of mercury. But by and large, these fish that we examined, reef fishes, really didn't have high concentrations. And this is not terribly surprising because, as I said earlier, this is often something we think of as affecting our more top predators, our larger fish, things like sharks or tuna. And so it really didn't turn out to be a big problem for the fish that we were looking at, which is great. Lead concentrations were high in the sediment at Kewalo, which is the harbor I showed you earlier, as well as Hanapepe Harbor on Kauaʻi. So both of those locations have the asterisks that I mentioned earlier. And the only fish that we saw with sort of problematically high levels of lead in their tissues was actually the state fish. And this was from Kewalo Harbor, or Kewalo Basin. And so it's not surprising, again, that we would see high concentrations of lead in fish from Kewalo, as it's a heavily urbanized, developed industrial site with a lot of runoff from roads and the sort of downtown, busy, bustling area of Honolulu. And so this was interesting and something that we definitely would like to look into further in the future. Looking at arsenic, which I discussed earlier as being high in the sediment from Maunalua and Hanapepe, as you can see again with the asterisks. What was really interesting is we saw quite high arsenic concentrations in a goatfish from Maunalua Bay. We also saw higher arsenic concentrations in goatfish that are near the Waimanalo Gulch landfill area on the west side of Oʻahu, as well as Hanapepe Harbor and Nawiliwili Harbor on Kauaʻi. And so these are all kind of large drainage outflow areas, but what's particularly interesting and what I kind of want to focus on here is Maunalua Bay, since that's something we'll be talking about more later. And the fact that we saw high arsenic in sediment that was sampled there, as well as in fish that were sampled there that do consume a considerable amount of sediment. Goatfish kind of comb through the sediment to get benthic invertebrates. And this would suggest that there is some degree of trophic transfer happening. When you see high concentrations in sediment and high concentrations in fish that eat sediment, this would suggest that sediment is a likely source of transfer. And so this is interesting and something that we'd like to look into further. Oh, again, these are just locations where we saw higher concentrations of arsenic in both the sediment and in the fish. Another metal that we looked at that was interesting was thallium. We saw high concentrations in Maunalua Bay in both the sediment. Again, you'll see the asterisk there, though we didn't discuss that explicitly earlier, as well as in fish, again suggesting a degree of trophic transfer. And what's interesting here again is that the locations where we see these higher the locations where we see these higher concentrations are Maunalua Bay, the Alawai, Nawiliwili Harbor, and these are all more urban areas, as well as Kewalo. And thallium was historically used as a rodent poisoning, a rodenticide. And so it's something that I'm curious about investigating a little bit more. Could this be linked to that in areas that are more urban? Could it possibly be from use of thallium as a rat poisoning? But that's not something we know for sure. Again, this is just some of the interesting stuff that you can begin to think about in terms of why do we see these higher concentrations in these locations. Oh, there we go. The arrows again are pointing to locations that have higher concentrations that are more urban. And next up is zinc and selenium. And so this is really interesting because basically we saw these really high, abnormally high concentrations in 4 nanui or chub that were collected from the same location near Hanapepe Harbor. And the same 4 fish had very high concentrations of zinc and selenium as compared to all the other fish that were sampled in this study. And so it turns out there are some really interesting connections between zinc and selenium in a more biomedical sense. And this is something that we're interested in looking at and sort of digging into. Why do we see these 4 individuals with such high concentrations as compared to all the other fish in this study? And is this something that's due to their diet or the location, or something that could be perhaps naturally elevated in this type of fish or in this environment? And so this is something we're looking into further, but is definitely of interest. And again, Honopepe is a really interesting location because it's a large harbor. It has a lot of agriculture nearby, as well as more urban and sort of industrial development. It drains a very large watershed. And it also has an area nearby that was a former dump or a place where people would bring all their garbage. There's also an airport nearby. And so there's a lot, it's a very sort of complicated and interesting location, and definitely warrants future investigation. And lastly, I want to touch on copper, because this is something that people worry about a lot as well in fish, because copper is used as an anti-fouling paint in harbors. And so it was interesting to see that all the locations that had the highest concentrations of copper, and again, I want to emphasize these aren't necessarily problematically high concentrations of copper, but just as compared to the other fish in this study, were all from areas with harbors. And so this would suggest that there is some relationship between the harbors and likely the use of copper as an anti-fouling paint and the concentrations that we're seeing in the environment, and specifically in marine life. While this might not be particularly problematic for fish or people at these levels, copper can be toxic to other marine life like corals and other invertebrates at very low concentrations. And so it's something to be aware of and to consider as we move forward when we think about how to manage some of these areas and things that we should be considering for the health of corals. And so to just sum up what we found in our past work for fish, I'll walk you through this image, which looks perhaps a little overwhelming at first, but it's not too bad. So on the left, we have all the different metals that we looked at, and on the right, we have the different fish species that we looked at. And I have this green bar here highlighting the lower trophic level fish. And basically, each line represents a link between the metal and the fish with the highest concentration of that metal in our study. And we looked at 150 different individual fish, and we tried to get roughly equal distributions of trophic groups from different locations. But we saw that we have the largest number of metals have the highest concentration in these lower trophic level fish like kole and manini and nanue and pualu. And so this is really was surprising, considering that, as I mentioned earlier, we often think of pollutants and metals as bioaccumulating and affecting the higher trophic levels most of all. And so while this isn't a definitive statement on the state of metals and fish in Hawaii, this was a preliminary sort of baseline study to help us get oriented as to what might be going on here. And it's definitely interesting and suggests that future research is warranted on these lower trophic levels to better understand how they may be affected by pollutants. And we do see our higher trophic levels, our fish that eat other fish, do have the highest levels, again, of things like mercury. And this is what we would expect. But it also is a helpful reminder that not all metals, not all pollutants behave the same. And so it's important to consider them independently when possible. But how do these findings translate to our work in Maunalua Bay? Well, as I said earlier, we're interested in Maunalua Bay largely because it's this large swath of reef that has these different input sources and these different types of potential pollutant transport. And so we're really interested in looking at these locations that receive input through either channelized streams or more natural free-flowing sources, or surface runoff, as well as the more heavily trafficked harbor areas. And so what we've done is collect sediment and fish at each of these locations. And this is all being informed by the past work that we've done at other locations around Oʻahu and Kauaʻi again. But this work is just more focused and more targeted on this smaller area, which we're sampling much more intensively to get a clear idea of what's going on in this area as a whole. And so in terms of the types of pollutants we're looking at, in the last work I talked about, we were really focusing on metals. But now we're branching out and we're expanding, and we're also looking at pesticides, PCBs, PAHs, things that are used for termites, as well as things we would expect from roads and urban areas. And so we're looking at a larger variety of pollutants that could be coming from the variety of sources that we might see in this very large watershed or set of watershed areas. And to do this, we're taking sediment samples in each location, as I said, but now based on our findings in the work I just presented, we're doing this in a different way where we're more comprehensively sampling with this multi-increment approach that you see on the top left, where we go out and take a lot of samples from an area and really thoroughly cover an area. And this has been in large part due to the help that we've received from the University of Hawaii OPIHI program crew. And so we had an awesome team of undergraduate students from a number of different universities here on Oahu helping out to enable us to do this very sort of comprehensive sampling approach. So I'm very grateful to them for all of their help with this portion of the project. We're also working with a number of organizations as well as community members in Maunalua Bay to collect fish samples from each of these locations. And this work has been slowed down a little bit by the recent events that have kept many of us in our homes. However, we are picking right back off where we left off. And so we're really fortunate to be able to work with a number of organizations that are helping guide us based on their experience working in Maunalua Bay and their relationships with people in this area. And so we're really grateful for their insight, as well as the help of community members that we've already received in this area. And this is allowing us to collect fish from these different locations that represent different trophic levels, as I said earlier. So we're looking at more predatory fish, fish that eat other fish, because we would expect them to have the highest concentrations of pollutants that bioaccumulate. We're looking at fish that eat a variety of different things, including invertebrates. And so these might represent a different component of the food web and a different way of being exposed to different contaminants. And because we're looking at a variety of contaminants now, they don't all necessarily behave the same. So it's important to get this wide spectrum of trophic groups. And so we're really excited to be able to include a variety of different types of fish. We're also including fish, again, that eat sediment, algae, and microorganisms. And this is informed by our past work, which suggests that these fish do end up with concentrations of pollutants that can be the highest of all the different types of fish in some cases. And so that makes them really important. And I should have said earlier, I forgot to mention that all of this work is really being guided by a desire to look at fish that people are interested in eating. And so whenever possible, we're seeking out fish that are of interest to fishermen in the area. And when that's not possible, we're looking for fish that would represent that trophic level and sort of give us an idea of what might be happening, and we can then use that to inform our understanding of other species. And what's really exciting is there's been a lot of work done in Maunalua Bay already. Recently, there was a study done in partnership with the University of Hawaii and the Department of Health, looking at water quality throughout Maunalua Bay. And all of these points on the map are places that they sampled. And so we're really excited to be able to contextualize our findings with other based on other research in the area moving forward. And this will let us offer a much more comprehensive view of what might be happening in this area. Because, for example, this study looked at different pollutants in water throughout the area, and now we're able to also contribute sediment and fish. And so this allows us to get a very robust understanding of what might be happening in these areas, as opposed to sampling more independently and disjointedly. And so we're really excited and grateful for all the work that has already been done in Maunalua Bay that can help us better understand our findings. And so, as I said, this work is ongoing. We were slowed down a little bit, but we are going ahead full steam, and we are really excited to be able to share this progress and sort of our game plan with you. And so stay tuned for more information. We're gonna be happy to share our results as we have them. And yeah, if you have any questions, please don't hesitate to contact me. My contact information is at the bottom of the screen here. And we're so grateful for the help of so many people who made this work possible. And so yeah, with that, I would just like to say thank you for tuning in. And again, please don't hesitate to contact me with any questions. And thank you for your time.